In this video, I'm going to cover some cool biological examples of allostery. I'm assuming that you've watched my previous video on allostery. If you haven't, you might want to go back and do that. But whether you do that or not, let's jump into this. So in the last video, we talked about these class of proteins known as GPCRs, which are receptors that will bind to a ligand outside or some molecule outside of the protein, outside of the cell, and then pass that information inside without that molecule actually going into the cell. And the basic way that they work is they sit in an inactive form in the absence of the agonist of the molecule that activates them. When that molecule binds, it triggers a change in the shape of the molecule, a change in its conformation, which then allows other proteins, these G proteins, to bind, undergo some functional transformations, and then go off into the cell and signal, hey, something bound at this GPCR. We're going to be what we focused on last time was this transition right here, right? How is it that the molecule goes from being in an inactive form when you don't have the hormone bound and into an active form when you do? How does the information go from I bound here to I change here? And we answer that question in terms of the idea of a linked equilibria, where the products of one reaction are the reactants to another. And so if we just think about the receptor itself, in the absence of any hormone, it sits in this conformation where it's favoring the inactive form. The receptor is off in the absence of hormone. If we add hormone, what that hormone does to this equilibrium is nothing directly, at least, right? Because it's on both sides of the reaction. The hormone isn't binding to either state, so it's just kind of there. But really, we see a change because this hormone does actually bind to the active form. And so as the concentration of hormone goes up, we're increasing the products of this reaction, which is going to shift the equilibrium to favor the reactant, by Le Chatelier's principle, right? So as this goes up, we shift this equilibrium in that direction. But as this equilibrium shifts towards the left, this equilibrium also has to shift to the left because we're consuming the A without the active form, without any ligand bound, or without any hormone bound. So we have to bring the inactive form up. And ultimately that means at high concentrations of hormone, we end up with all active bound form. So we can write that out sort of in a set of steps. So first, adding X adds excess products to this reaction. So we've got more products to this reaction here. So that's 1. The equilibrium shifts in response, so we start seeing the formation of more complex. That's 2. That has the effect of depleting A, right? So the A concentration starts to go down. That's 3. And to compensate for that, we start seeing I shift into fill in for A, basically. So we see the equilibrium shift in response. So this is just Le Chatelier's principle, right? We link two equilibria, we make the products and reactants of these reactions the same, and that means that if we change one part of the reaction, in this case, we start binding an X to the active form, we shift the entire equilibrium towards that active form. And the reason that this works is because and actually passes a signal is because this active form in purple binds to both the hormone and the downstream target, right? So when it binds to the target, it activates because it opens up the spot to bind to the target. So which state binds to which target actually helps to set how these receptors work and what the logic of these systems are. So we're going to talk about 3 different variations on this basic theme today that we see biologically. The first one being more of sort of theoretical, like this is the sort of thing we might see in the exact way we've been talking about the GPCRs. And then the other 2 are some very sort of cool, very specific systems that do specific things. So first, let's talk about the regulation of enzyme activity. So the GPCR example, we weren't really regulating an enzyme. We were actually just changing the shape of a receptor protein. But it turns out that allostery can directly regulate enzyme activity. So here, for example, is an enzyme. Here's, for example, is an enzyme that is an equilibrium between an inactive and an active form, where KEQ is greater than 1. So this is actually a good time to practice your biochemical reasoning. So if KEQ is greater than 1, what does that mean? And would we see any enzyme activity if we just looked at the protein in solution? So if you'd like, you can pause and think about that for a second. What does KEQ greater than 1 mean? And would we see enzyme activity? All right, you had your chance to pause. The answer is that KEQ greater than 0 means that E active is favored, right? That at equilibrium, we're going to get a higher concentration of active than inactive, which means that we, in fact, would see enzyme activity, right? Because the active form of the enzyme is populated. Okay? Now we're drawing an equilibrium, and in that equilibrium, what we have is something like this, where E inactive is bound to X, E inactive with X here, E active with X here. So what's going to happen to the enzyme activity we see as we add X? So again, this is a good chance to pause and think about it. So what happens to enzyme activity as we add X? All right, I'm assuming that you came out of your pause, and the answer is that we are going to lose activity. The addition of X is going to lower the activity of the system. And let's unpack this with another one of these examples here. So what we're going to do is we are adding X. X makes excess products for E inact X, right? So we're making excess products here. What that means then is that we're going to shift to E inactive X. So we're going to shift towards this state that will deplete E inactive by itself. So it's going to deplete this molecule, which is going to cause for E active will become E inactive. And so what we're seeing here is the addition of molecule X can cause an enzyme to turn off. So that's something, it's modulating the activity of the enzyme. And it's also doing so without actually binding to the active form, right? This isn't sort of competing, isn't sticking on the active form anywhere. It's binding to an inactive form of the molecule and shifting it this way. And so what this demonstrates is two things. That one, allostery can regulate enzymes. So we can turn enzyme activity on and off. And two, does not need to bind the active form. In fact, in this case, what's happening is the X molecule is binding to the inactive form, and that's changing the activity. Okay, so with the GPCRs, we saw how binding of the molecule turned on the system. Now we're seeing binding of the molecule turned off the system. So that's one sort of variation. And just at a theoretical level, I wanted to show that so we can see, oh, the logic of this doesn't always have to go bind and turn on. It can also go bind and turn off. Now I'd like to talk about two biological variations. One is phosphorylation. So what is that? So phosphorylation is when a kinase, this is an enzyme, covalently attaches a phosphate to a specific amino acid on a protein. And we could draw this way. We have a target protein coming in here. The kinase is causing an activity in the middle where it's taking a phosphate off of ATP and sticking it onto the target. This usually occurs at threonine or serine, and it occurs at specific residues. So it's not just any serine or any threonine. It's like the enzyme, the kinase, will put a specific serine on a protein. It will take that hydroxyl, which is the normal side chain of a serine, and it will turn it into a phosphate. It'll put a phosphate on instead of that hydroxyl. Okay. Phosphorylation. What does that do? Well, let me show a real example of this. So I'm going to talk about the enzyme MAP4K1, and it helps pass signals from the cell surface to the nucleus. So there's a class of receptors that bind things, just like GPCRs, although these are not GPCRs, they're a different class of receptors. And then what it does is it passes information down using these MAPK enzymes. And so MAP4K1 is activated by phosphorylation. So it gets phosphorylated, gets a phosphorylated site stuck onto it. And then when it's activated, it phosphorylates downstream targets. So it starts attaching phosphates to things downstream of it. So this entire thing is actually often known as a phosphorylation cascade because the signal upstream signals downstream phosphorylate, 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 and ultimately that ends up triggering, in this case, the production of target genes in response to the extracellular signal. Okay, cool. So that's the biology. How does this phosphorylation translate and work mechanistically for allostery? Well, here I'm showing the structure of this protein in the inactive form, and I'm highlighting the phosphorylation sites, the threonines that are the threonines and serines that get phosphorylated in orange, and then the active site residues, the place on the enzyme where the chemistry actually happens in blue. And hopefully what you can appreciate is it's all kind of jammed in the middle. The active site is buried, the phosphorylation sites are actually buried, so it's all kind of jammed into the center of this protein. If we look at the phosphorylated form of the receptor, hopefully what you can, the enzyme, not the receptor. The phosphorylated form of this enzyme is much more open looking, and hopefully you can appreciate that things like the active site, I'm going to pause. This active site is actually kind of accessible now, right? I can imagine a small molecule getting in there and something happening. So in the unphosphorylated form, the active site is buried. In the phosphorylated form, the active site is exposed. And this is a pretty big structural rearrangement to get there. So now I'm showing a morph between the two structures. And hopefully what you can appreciate, I've sort of fixed the blue. I've kept that in the same orientation. But the gray structure is flipping over. It's actually flipping 180 degrees as phosphorylation happens. And that is what exposes the site. So the site is buried here, and then now it's exposed, right? It's open and accessible. It's now, when we go back to the unphosphorylated form, it's buried. And now when it's phosphorylated, the active site is exposed. And this is driven actually by a negative charge on the phosphate. So here are the phosphates in the active form. They're all happily exposed to water. A phosphate is negatively charged, so we get a bunch of negative charges. But this is okay because they're happily interacting with water molecules. So those wedges are waters with their oxygen at the tip and the hydrogens at the bottom. But imagine we put negative charges down here in the inactive form, buried away from water. What happens when you put a negative charge away from water? Well, it really would rather interact with the water than be in a negative state. And the way that this system deals with that is, okay, negative charge, well, why don't we just rotate the gray subunit, expose these to solvent, and expose these to water, and then expose the active site. So the negative charge on the phosphate drives this conformational change. What this then does is it exposes this active site and turns on the enzyme. So this enzyme, by getting phosphorylated, by getting these things bound at these sites, which are fairly distant, they're not anywhere near the actual enzyme active site, transmits the information effectively, hey, you should turn on your enzyme. You should turn on the enzyme just by phosphorylation at these sites. Okay, so we've now seen a couple of different examples where binding somewhere, or now in this case phosphorylation somewhere, transmits to another location of the protein. The last example I wanted to talk about is actually a multi-input system, where instead of just having binding or just having a phosphorylation, it actually has both. And the example that I want to talk about is this thing called AMP-K. AMP-K is a high-level metabolic regulator. And so this is from a recent review where basically they said, here's the AMP-K, and here are all the pathways that it regulates. You're probably looking at that saying, I can't even read that. There's so many things going on, and that's true. It's a very complicated bit. I'm just going to point out two things, though. When this gets activated, it goes down here. It activates this pathway, it activates this pathway, and it activates this pathway. Fatty acid oxidation. That is the production of energy from breaking down fats. And then if we look up at this one, what is it doing? It's doing this, glucose oxidation. It's saying, let's make energy from sugars. So it's saying both make energy from sugars and make energy from fats. So it's a high-level metabolic regulator that's going to activate energy production. Okay, cool. The way that this molecule works is it's a heterotrimer. So it's three different proteins that interact together. A gamma regulatory subunit in blue, an alpha kinase subunit in green, and then a structural subunit in purple. And what I'm going to show in the next slide is the difference between the inactive form and the active form. So here is the inactive form of this. I want to note a couple of things. It has a phosphorylation site that's actually sticking off into water. It has an active site, but this is actually buried. It's very difficult to get molecules in and out of that active site. And then finally, is this regulatory domain has ATP molecules bound. So when ATP concentrations in a cell are very high, that's basically saying there's plenty of energy around. Don't turn on pathways that we need to release energy, right? So this is saying keep this molecule in the off conformation. The active form, by contrast, is this big open thing. Let's look at the active site first. The active site is here, and hopefully you can appreciate that that's actually more accessible. Maybe not without looking at the structures, but this is more accessible. So this is actually able to do its job. This molecule is being activated. Its active site is active when it's in its active form. We have AMP bound up here now, and that is adenosine monophosphate. So it's an ATP that's actually lost two phosphates. So this is a strong indication that the cell has a low energy state. We need to start doing things like breaking down fats. We need to start doing things like burning sugars. So that is allosterically activating. It's actually the binding of that molecule that takes this blue subunit and puts it up here into this top form, right? So the motion of that subunit is driven by binding of adenosine monophosphate. And then the last thing I want to point out is this phosphorylation site's actually here in the protein. It's gone from being totally interacting with water to being inside the protein. You might say, well, it's bad being inside the protein, but actually what's happening is the AMP is stabilizing an active form. It's putting it into this state here. And then the negatively charged phosphate actually interacts favorably with a positively charged lysine. So when we put a negative charge on that phosphate, on that threonine, you get a nice interaction with the positive charge, even further stabilizing this active form. So AMP is stabilizing the active form. That's an indicator that cell energy is low. And then there's a second gatekeeping phosphorylation event that's also stabilizing the active form. And these two things together say, okay, go ahead and start breaking down fuel. And so what we have is this thing. This is a kinase that phosphorylates AMPK. This is basically turned on by a variety of other upstream pathways. It's going to get very complicated if I go into the full biology of that, but this is basically a checkpoint protein that is on to make sure that AMPK can be turned on. AMP binds to AMPK, so it binds up here and shifts it into the active form. And together, these actually turn the molecule on. So let's think about it this way. When we have this molecule around, or not around, we could think about it, there's two possibilities. It's either not around or it's around. And we can think about with AMP, it's either not around or it's around. And so this is all binary combinations of those. So if it's neither phosphorylated nor AMP around, it's going to be off, right? And if it is phosphorylated but there's no AMP, it's going to be off. If it's not phosphorylated and there's AMP, it's going to be off. But if you have both of these together, phosphorylation and AMP, it's now on. So from a logic perspective, these operate with AND logic. So now we're adding two layers of information. It's not just, oh, something bound, turn on. This is now, if something binds and you're phosphorylated, turn on your molecule. And this is, just the beginning. If we kept going, we could find all kinds of crazy ways that different biomolecules operate with AND logic or maybe OR logic, anyway. So they're all going to interact to give rise to this underlying behavior that cells do, which is computing information or information processing. Okay, so what should we take away from this? First, allostery is when the change at one site alters the activity at another, and this allows proteins to, say, respond to signals. Allostery happens all over. I chose this set of examples just somewhat randomly. I mean, I could have chosen a whole bunch of these because these occur all over the place. And allostery has a variety of different possible things it can do. It can activate, it can deactivate, it can be a covalent bond, say like a phosphorylation. It can be non-covalent binding, and allostery can even take multiple inputs to give rise to specific outputs. Okay, so that's what I have on sort of these different ways that allostery works. In the next video, and that's the last in this mini-series, we're going to talk about cooperativity, which is sort of a slightly different form of allostery, but that leads to some very cool and important ways that cells can go from, say, being sort of on to all the way on in a big sort of step function sort of way. So I look forward to tackling that with you, and thanks for listening, watching, whatever it is you're doing, whatever the right verb is. So see you later.